Vamos a presentar a nuestro siguiente speaker, es el doctor Vasconcelos. Él actualmente trabaja y vive en San Pablo, es un médico formado en de oftalmología y ha realizado un fellow de clínico y quirúrgico de retina y vitrio en la Universidad Federal de San Pablo, la UNIFES, y posteriormente realizó su fellow de genética ocular y terapia génica en Casey Eye Institute en Portland, Oregón. Y bueno, actualmente forma parte de la Retina Clinic y del Instituto de Genética Ocular en San Paulo. Así que Uber, un gran amigo y mejor oftalmólogo. Así que adelante, Uber, con tu presentación. Muchas gracias.
Gracias, Joan. Gracias por la invitación. Yo voy a share my screen here. So I will present it in English since there are some terminologies that I think it will be better for me to present in this language. Thanks for the invitation, and I will talk a little bit about the concepts of gene therapy. So gene therapy refers to the treatment technique that modifies, substitutes the genetic material of the receiving cell by altering its transcribed biological material and its functioning. And the classical form of this therapy refers to the introduction of an external genetic material into an organism for therapeutic purpose. Other forms of gene therapy will be presented during this talk. In most of cases, for the realization of this therapy, it's essential to know the molecular mechanisms and the genetic alteration that is directly linked with the treated disease. So in this case, the altered gene will be the primary target of the therapy. Other strategies are genetic treatments to increase the production of a beneficial factor, or even editing the gene that we'll talk a little bit about later. The gene can be introduced in the target cell into two ways, in vivo or ex vivo. In the in vivo technique, the introduction of the gene occurs directly in the organism, while in the ex vivo technique, the cells are removed from the individual, genetically modified, and then reintroduced. In the ocular gene therapy, the in vivo technique is currently used, while the ex vivo technique has been applied more in preclinical research and even in treatment of some oncology problems like lymphomas. During these years of evolution of gene therapy, the world of inherited retinal disease has also changed. To get into what we currently have in the scenario of eye gene therapy itself, first and very important is to determine what we are dealing with. So in all times, we are referring to disease in the retina and in the eye by a broad name like retinitis pigmentosa, Stargardt disease, Leber congenital amaurosis, Usher syndrome, and other ones. And nowadays, we know that these genetic changes are caused by a specific pathogenic variation, with specific pathogenic variations in specific genes, and that genes can cause these phenotype presentations. So it's very important for us to get inside this new world and get inside the genetic changes that can cause these old known phenotypes, so we can individualize patients and be able to treat these patients regarding the genetic change that this patient has. And how was the evolution of gene therapy? Since the DNA model was described by James Watson and Francis Crick in 1950, the gene therapy started to be some kind of reality. In the 60s, have some kind of proof of concept about a possible genetic transformation by exogenous factors. And the first ex vivo proof of concept experiments were done in 70s. And then in early 90s, that come into clinical trials already in humans. And at that time, we had some high toxicity problems that even lead to the death of a patient in one of these trials that was trying to treat transcarbamylase ornithine deficiency. And that was like a pushback on the gene therapy evolution at that time. And we tried to do more research, lab research, focus on basic science. But then in early 2000 and in the end of the 2010, with the development of the adeno-associated viral vectors, and then later with more clinical trials and with some safety modifications and improvements in the gene delivery, we came back very strong on the gene therapy, and that come back to the center of our attention. The gene therapies are being investigated in different diseases, since hemophilia until wet and dry AMD, and that is becoming more and more reality nowadays. For most gene therapies, one of the most important aspects concerns the vector that will transport the genetic or biological material to the target cell. The ideal vector should have low toxicity, high homogeneity, and the ability to express the transgene in a stable level in the cell, low cost. And nowadays, the most used ones are lentivirus, adenovirus, and the adeno-associated virus, the AAV, with the lentivirus and the AAV being used in some clinical trials in ophthalmology. The AAV is the one that is most used with a lot of pros, with very efficiently reach the retina, with able to transduce the target cell, very reliable and safety viral vector, and with some cons that is the capacity of carry a very a small amount of gene, and that goes to 5 kilobytes, and that make it unable to carry some kind of genes like ABCA4, myosin 7A, between others, and then the lentivirus can be an alternative in that spectrum of problems. About the treatment routes that we have for this disease, in ophthalmology we have two main forms of treatment, subretinal injection and intravitreal injection. These routes allow lower drug volume, higher rate of transduction, and lower rate of complications due to this restricted drug effect. Other kind of route treatments like topical, intracameral, intrastromal routes is still on test and being tested on animals and other preclinical research. About the gene therapy itself and the viral vector that you talk about, and subretinal injection and the intravitreal injection, we'll have some types that the gene will be changed in our organs. The main one is the gene augmentation therapy, and that is the type that we put a genetic material in our body, and that's the main one when we are talking about gene therapy. The other ones, I use this main classification here together in the types of gene therapy, but actually they act in different ways, but make change in the genetic. And that includes gene editing, antisense oligonucleotides, and nonsense bypass therapy. I mark these ones mostly because they are the ones that we have clinical trials nowadays in ophthalmology diseases. So we'll talk a little bit more about it. Here I put a list of some of the clinical trials that is still active today in 2020. There is some that is coming and getting active too, for all of the genes using all types of that virus vectors that we talk about, and in different phases of protocol, coming from ABCA4, causing Stargardt disease, from CEP290 and RPE65, causing Leber congenital amaurosis, to CHM, causing choroideremia, and other diseases like achromatopsia and retinitis pigmentosa. And about the types of gene therapy. First one, the gene editing, is one that the DNA of the target cell is directly altered by the insertion, deletion, or replacement of a specific DNA sequence. This type of gene editing can be done by zinc finger nucleases, TALENs, that's some kind of proteins that can actually do this editing. But the main form of this gene editing is the CRISPR. The CRISPR-Cas9 system is related to a Cas9 protein and a guide RNA. And this CRISPR can get and modify the genetic sequencing that is the target for it. Currently, the CRISPR-Cas9 is the most widespread and studied method of gene editing. And we have protocols, clinical trials actually done in ophthalmology, and I'll talk about it later. About the antisense oligonucleotides, they use like an RNA oligonucleotides small sequencing that creates some kind of bridge in the pre-RNA messenger, the messenger RNA, and when that is translated to a protein, the protein is altered from that protein that was with some kind of disease to a new protein that it's actually closer or even equal to the normal protein that can be functional on this patient. Another type of gene therapy treatment is the ones that treat the nonsense mutations. The nonsense mutations usually generate dysfunctional proteins, and it's very common to see this kind of mutations in all kinds of inherited retinal disease, including choroideremia, for example. And this nonsense bypass therapy, they create they are read-through drugs that make the translation and the interpretation of the RNA able to go through these nonsense mutations. This kind of treatment, there is some clinical trials active in choroideremia using these drugs here, atelurane, and then I'll get PTC414. To finish, the gene augmentation therapy. That is the main form of gene therapy in humans to date. Through the addition of a genetic material in the target cell, there will be an increase in a protein expressions for various biochemical purposes. So why we use this technique? There is, for example, disease with biallelic mutations in some genes that the protein is absent in in his action. So what happens is that you put like a genetic material that will be translated in our cells, and that will produce that protein that is lacking in our organs. Other case that we can use that is like when the protein is insufficient in our organism, so we can increase the production of this protein using this technique. Here we have a gene therapy surgery, gene augmentation therapy surgery. That was a surgery done in a patient with biallelic RPE65 mutation with the first drug approved for this kind of treatment, that is the voretigen neparvovec, that treats this kind of mutation in this gene, that is the RPE65, and can cause Leber congenital amaurosis. As you can see, we do a subarachnoid injection of this drug, and in the target areas that you have available rod cells and we can treat the cells and increase the production of the RP65 in this patient that has lack of this protein in their organism. So an important information is that this gene therapy actually was approved to be done in Brazil, following the way that was done in other countries. It was approved in the US in the end of 2017, in Europe in 2018, and now last month was approved to be done in Brazil. It's still in the process of specifying the drug, but actually we are ready now to do surgery here in Latin America. And about the future, like I told you, the CRISPR-Cas9 gene editing therapy was done first time in a human body in an ophthalmology clinical trial, and that was done on OHSU, where I stayed there doing my fellowship in gene therapy and ocular genetics. And that is the future. We still ongoing developing new forms of treatment regarding inherited retinal disease, and even more each day that will be a reality for us in retina special in all ophthalmology. I want to thank you again for the invitation. Here are my contacts, and feel free to ask any questions that you want. Thank you.
Excellent, Uber. Excellent. Great talk. Congratulations. I have some questions for you. Vou falar em portunhol, porque eu sei que você gosta de portunhol e pode treinar o portunhol aqui com nós. Qual é a sua opinião sobre o uso de OCT intraoperatório nesses casos? Eu acho que talvez você queira falar sobre se verdadeiramente pensa que é muito importante para colocar ou assegurar o sítio onde estamos botando a terapia gênica diretamente. O que você pensa disso?
Sim, sempre quando falamos com os especialistas de retina sobre a infusão subretiniana de alguma droga, todos falam, sim, temos a experiência com os TPAs, que isso para hemorragia subretiniana. É pouco diferente acá, porque quando tem a hemorragia e está tratando com TPA, normalmente a retina já está descolada, então é pouco mais fácil de fazer o procedimento. E acá estamos falando de drogas que custam realmente, nos Estados Unidos, custam $425 mil por ojo tratado. Então, a precisão do tratamento e onde vamos a fazê-lo é muito importante. Então, o OCT intraoperatório não é essencial, como falei, pero é importante porque ele pode mostrar algumas características da cirurgia e alguns problemas intraoperatórios que vimos nesse tipo de cirurgia. Eu publiquei esse ano artículo sobre os achados de OCT intraoperatório neste tipo de cirurgia. Então, tem algo que o OCT pode agregar, e por ser tratamento muito caro e muito preciso, pode ser sim uma maquinaria necessária, mas não essencial para o tratamento.
Perfecto. Muy bien, muchas gracias. Y